You are holding a sealed tube of cloudy water. It was drawn from a salt lake in eastern California, a lake so saturated with arsenic that almost nothing should be able to survive in it. And for a short stretch of time, a significant part of the scientific world believes that what is swimming in that tube is a second kind of life, not a new species, a new kind. It is not. But the reason it is not turns out to be far more interesting than the claim ever was. Because life could be fundamentally different from us in exactly 4 ways. 3 of those differences would be found on other worlds. The 4th would not be found in space at all. And underneath all 4 sits a question nobody has answered. Would we recognize life if we were already holding it? Biology looks enormous from the inside. Millions of cataloged species. Organisms living in boiling water, in acid, in solid rock 2 miles underground. It is easy to look at that range and conclude we have seen what life can do. We have not. We have seen what one lineage of life can do. Every organism ever studied, without exception, runs the same core chemistry. Water is the liquid that everything happens inside. Carbon is the structural backbone. Phosphorus holding the genetic material together. Proteins assembled from the same small set of building blocks, every one of them in the same orientation. A genetic code that is effectively identical from bacteria to blue whale. That is not variety. That is a single design, elaborated for 4 billion years. All of it descends from one ancestor, which means the sample size of biology is 1. Imagine trying to describe every language on Earth after overhearing a single sentence. You could describe that sentence perfectly. What you could not do is say which of its features are required for language, and which are simply how that one sentence happened to go. That is the position astrobiology has been in from the beginning, and it carries a hard, practical consequence. Every instrument ever built to search for life encodes an assumption. It assumes alien life resembles us. Look for water. Look for carbon compounds. Look for the molecules our own chemistry produces. If life out there is built differently, the instrument does not report a mystery. It reports nothing. The sample is filed as ordinary chemistry, and the search moves on. To understand how badly that can go, it helps to look at what happened the one time a research team believed they had found the second sample. Not on another planet, in a lake. The lake is Mono Lake in California. Its water is salty, alkaline, and loaded with arsenic, an element that kills most living things by imitating phosphorus closely enough to get inside a cell and then failing to do phosphorus's job. A researcher named Felisa Wolfe-Simon, then with the NASA Astrobiology Institute, working alongside Ronald Orumland of the United States Geological Survey, went into those sediments looking for something specific. Not a tough organism, a different one. She wanted to know whether life could be built using arsenic where every other living thing uses phosphorus. She isolated a strain of bacteria and reported that it could do exactly that. Grow on arsenic, take it up into its own genetic material. 6 elements had long been treated as non-negotiable for life, and she was proposing that one could be swapped out. The strain was designated GFIG-1. The letters stand for Give Felisa a Job, a reference, half joke and half not, to her long search for a permanent research position. The finding was announced at a press conference. One of her co-authors was Paul Davies, a physicist who had already published on the possibility that alternative forms of life could exist and go unnoticed. What happened next is the part worth paying attention to. The field did not dismiss it. The field tested it. A microbiologist named Rosie Redfield at the University of British Columbia spent months simply learning to grow the organism reliably. Then, with Marshall Reeves at Princeton, she ran its purified genetic material through an instrument that weighs molecules precisely enough to identify which atoms are inside them. There was no arsenic bound into it. Separately, a team led by Tobias Erb and Julia Vorholt in Zurich demonstrated that the bacterium survives by scavenging tiny traces of phosphorus out of its surroundings. Redfield's own account of why she bothered has stayed with me. She said she agreed with everyone else that the result was almost certainly wrong, that trying to replicate it was close to a waste of her time, and that she was going to do it anyway, because somebody should. Recently, after roughly 15 years of argument and failed replications, the journal retracted the paper. No fraud was found. No misconduct. The original authors still disagree with the decision, and the organism is real. It turns out to be extraordinarily resistant to arsenic, which is a genuine and interesting finding about a genuinely tough bacterium. So that is one of the 4 ways life could differ from us. Different building blocks. Tested carefully and so far not found. Here is where it gets strange. The other 3 have barely been tested at all. When most people imagine alien life, they imagine shapes. Tall, short, gray, insect-like. Shape is the least interesting thing about it. Shape is just what evolution does with whatever chemistry it was handed. The real differences sit underneath, and each one is independent of the others. The first is the liquid. Every chemical reaction in your body takes place dissolved in water, and water is doing specific work in there. It carries charge. It holds molecules in shape. It stays liquid across a wide and useful range of temperatures. The second is the elements. Which atoms build the structure and run the machinery? That is the axis Mono Lake was testing. The third is handedness. That one is the strangest, and I will come back to it. The fourth is the origin. Not what life is made from, but how many separate times it started. Start with the liquid. There is exactly one other place in this solar system with stable liquid on its surface, and it is not a planet. It is Titan, the largest moon of Saturn. Titan has weather. Rivers, lakes, and seas, under a thick orange atmosphere you cannot see through. What it does not have is surface water in liquid form. It is far too cold. On Titan, water ice is bedrock, hard as granite. The liquid filling those seas is methane and ethane, the same compounds we burn as natural gas here, held liquid near 180 degrees below zero on the Celsius scale. The obvious objection is that no chemistry interesting enough to be called alive could run that cold. But a team at Cornell narrowed the question down to something testable. A graduate student, James Stevenson, working with the planetary scientist Jonathan Lunine and the chemical engineer Paulette Clancy, set aside metabolism and genetics entirely and asked one thing instead. Could you even build a cell wall in liquid methane? Every living cell needs a boundary, something that holds the inside in. They modeled one. They built it in simulation out of a simple nitrogen compound that radio telescopes had already confirmed floating in Titan's atmosphere, and they called the structure an azotosome, a cell membrane assembled from nitrogen compounds instead of the fats our own cells use. In the simulation, it held together, flexible, stable, roughly the size of a virus. And then, as science does, somebody checked. A thermodynamic analysis out of Sweden argued that the structure could not form under real Titan conditions at all. That was still theory, arguing with theory. The first physical experiment came recently from Tuan Vu and Robert Hoddes at NASA's Jet Propulsion Laboratory. They took the compound, put it into liquid methane and liquid ethane at Titan temperatures, and watched what it actually did. It did not build membranes. It locked together with the ethane into a solid crystal, not a cell wall, a mineral. Their own paper is careful about the limits of that result. It does not rule out structures that form briefly and fall apart again, and a different route to building membranes there has since been proposed. But the most attractive version of life in a liquid that is not water was finally tested in a laboratory, and the laboratory said no. Now the third difference. This is the one that unsettles me. Hold your hands up in front of you. They are the same hand. Same fingers, same joints, same structure. And they are not interchangeable because one is the mirror of the other. And a left hand does not fit a right-handed glove. Molecules do this too. Most of the molecules life is built from come in two mirrored versions, identical in every measurable property except their orientation. Chemists call this property chirality, which simply means handedness. Here is the fact that should stop you. When you produce those molecules using ordinary non-living chemistry, you get both versions in equal amounts, 50-50, every time. There is no energy difference between them. Nothing in physics prefers one over the other. But life is not 50-50. Life is absolute about it. The proteins in your body are built almost entirely from left-handed building blocks. The sugars threaded through your DNA and your RNA are right-handed. Every organism on this planet, without meaningful exception, uses exactly the same handedness you do. Nobody knows why. There are proposals. Polarized starlight in the clouds where planets are born. A vanishingly small asymmetry buried deep in particle physics. Simple chance, locked in early and never revisited. None of them is settled. Now consider what a mirror organism would be. Same idea, opposite hand. Right-handed proteins, left-handed sugars. It would be chemically ordinary. It would obey every law of physics we know of. And it would be very close to invisible to us, because our detection methods work by having our molecules recognize the target, and our molecules would not fit. The test comes back clean, not because nothing is there, because nothing there is shaped to be caught. There is one useful inversion buried in this. Since non-living chemistry produces both hands in equal measure, a sample that is lopsided, heavily one hand and not the other, is one of the very few signals that points to life without assuming anything about which molecules that life uses. Which brings us to the stakes. Every argument about how common life is in the universe rests on a single unknown. Whether starting life is easy or very nearly impossible, we have one data point, and one data point cannot distinguish between a process that runs readily wherever conditions allow, and a fluke that has happened once in everything we can see. But if life started twice on the same planet, independently, that question is settled. Two separate beginnings on one small rocky world would mean the process is not a fluke. It would mean that anywhere the conditions are met, it probably runs. And you would not need a telescope to establish it. A single organism here with an origin unrelated to ours would tell us more about the rest of the universe than every exoplanet atmosphere measured so far. The reverse carries weight too. A serious, thorough search that comes back empty would tell us something real about how rare our own beginning was. So here is the fourth kind, the one that is not in space. The idea was named formally by Cleland, a philosopher of science at the University of Colorado, working with the biochemist Shelley Copley, and developed further in a paper with Paul Davies, Stephen Benner, and others. They called it the shadow biosphere, a second, independent origin of life still living on Earth right now. The proposal is straightforward and difficult to dismiss. If life arose here once, under conditions we know existed, there is no obvious reason it could not have arisen more than once. And if it did, the descendants of that second beginning could still be here. Microbial, unrelated to us, sharing the same planet, the same rocks, the same water. So why has nobody found them? Because of how microbiology is done. To study a microbe, you generally have to grow it, and you grow it in a broth designed around the chemistry of the life we already know. An organism running different chemistry sits in that broth and does nothing at all. It is not that the search failed. The search was never aimed there. Cleland has pointed to one specific place worth checking. Desert varnish, the dark, hard coating that forms slowly on exposed rock in dry places, whose origin has been argued over since Darwin was alive, and which nobody has been able to reproduce in a laboratory, biologically or otherwise. She has noted with some frustration that nobody has yet gone and looked. And underneath every one of these 4 possibilities sits the problem that makes them possible. We have no general definition of life. What we have is a detailed description of ourselves. So every instrument we build, every test, every probe sent anywhere, is calibrated to detect the single example we already possess. There is one more thing worth saying, because this is not a counsel of despair. There is a serious effort underway to fix exactly this. NASA funds a research program built around detecting life without assuming its chemistry, led by Sarah Stewart Johnson at Georgetown University, with Heather Graham at NASA's Goddard Space Flight Center as her deputy. The approach is to stop hunting for our molecules and start hunting for patterns that non-living chemistry does not produce. Lopsided handedness. Molecules too complex and too repetitive to be accidents. It genuinely widens the net. What it does not do is tell us what life is. We tend to frame the search for life elsewhere as a problem of distance. Bigger telescopes, longer missions, better sensors, more patience. It is much closer to a problem of self-knowledge. We are trying to recognize something we have never managed to define, using instruments calibrated to the only example we have ever seen. Those 4 differences are not exotic possibilities. They are the 4 ways our definition could be wrong. And the question worth sitting with is not whether life exists somewhere else. It almost certainly does. The question is, what would have to be true of something before we agreed to call it alive? Not similar to us. Alive. Because the strangest possibility here is not a mirror organism drifting in a methane sea more than 1 billion kilometers away. It is that the second sample has already been collected, scraped off a rock, sealed in a tube, run through a machine calibrated for our chemistry, and recorded correctly as nothing.